Hello, let's discuss on transcription unit. What is a transcription unit? A transcription unit is that segment of the DNA template that transcribed and form RNA. So here, if you look at the diagram, there are two lines indicated, one with blue, one is with red. So this blue strand is called as the template strand of the DNA running from 5 towards 3 direction with the arrow, and this is the coding segment. This red color is the coding segment. This is running from 3' towards 3', but both of these are running in opposite direction, that is anti-parallel strand. Now, a transcription unit consists of 3 regions. The first one is the promoter, the second is the structural gene, the third is the terminator. So let's first discuss on the promoter region. The promoter region is the portion of the DNA where the RNA polymerase gets bind or come into contact with the DNA so that the process of transcription can be started. And this promoter region is located in the upstream, this side, upstream of the initiation codon. We shall be discussing upstream, downstream later on. So promoter is the region where the RNA polymerase come and binds the DNA to start the transcription. Now the second is the structural gene. This is the structural gene. Structural genes is the gene that code for RNA. So this structural gene is present only in the template strand of the DNA. This structural gene is the gene that transfer the genetic information to the RNA, and hence this template serve this region of the DNA serve as a template for the synthesis of particular amino acid. Therefore, this structural genes is also the functional unit of the inheritance. That means the genetic information are being transferred from this template strand of the DNA. The coding strand of the DNA, that is this strand, doesn't get transcribed during transcription of RNA, and it carries only the nitrogenous base that is complementary to this strand. So we know that if this side is A, this side will be T. So this region, this strand of the DNA will be carrying the complementary base sequence for this template DNA. Now, during transcription, let's look at this diagram. This is the template, this is the coding. Now during transcription, before transcription, the DNA will get unwind, will be getting separated, and the RNA will be coming to transcribe this template, not this one, because this is the strand of the DNA that will pass the genetic information. So the RNA will come, and the template, if it is carrying nitrogenous base A, T, C, G, the RNA will be carrying the complementary sequence. But in case of RNA, it will be this. We know that A always pair with T, but in this case we know that in RNA the T is being replaced by U, that is uracil. Therefore RNA will give U. Therefore A will pair with U in case of RNA. In case of DNA, it will pair with T. Now T will pair with A, C with G, G with C. So this if we look at the genetic information that is being transcribed by this RNA, and if we look at the complementary strand of the coding segment of this DNA, then genetic information, the nitrogenous base of this transcribed RNA and this coding region is same, except in case of RNA it is T, and in case of coding segment of the DNA it is T. So the coding strand of the DNA will be same as that of the transcribed RNA, except that in case of this coding strand it will be T, and in case of RNA it will be U. So the coding strand will be carrying the same nitrogenous base as that of the transcribed RNA. Now this is the second region of the transcription unit. The third is the terminator region. This is the region where the transcription process stops, and this is located in the downstream. Now let's discuss on what is downstream and what is upstream. If we look at this diagram, we can see that the transcription is taking place in the template region only. That is in the 3. This template is running in 5 to 3 dash strand, and the non-coding region. This is the non-coding region is running in 5 dash 3 dash. In both of the strands are running anti-parallel. When we talk about the promoter and the terminator region, we are only referring to this coding segment of the DNA. So if we look at the promoter region, we find this promoter region in the 5 dash strand of the coding segment of the DNA. Therefore, we call this promoter as the upstream. This 5 dash, this called as the upstream, and the terminator region is located in the 3rd dash, towards the 3rd dash polarity of the coding strand. Therefore, we refer this terminator region as the downstream of the transcription unit. So this is all about the transcription unit.